Hello, class. Welcome to our first lecture. So in this opening section, we're going to set a foundation on what evolutionary psychology actually starts out with. And the foundation of that is natural selection. You've probably studied natural selection before, but just to make sure that everyone is on the same page, we're going to start with a basic definition. The definition of natural selection is that replicators that cause their own reproduction better than alternatives will come to predominate in the population. Now, there are a few terms there and moving parts like replicator, reproduction, alternative. Let's make sure we understand what those mean. So a replicator is anything that, by virtue of its interaction with the environment around it, reliably makes self-copies that inherit that ability to make self-copies. An alternative is any alternative design that is a similar kind of replicator, but instantiates a different effect and therefore has a different heritable basis. Let's look at a few different examples of how natural selection works so that we can best understand how we can make predictions about how it should work in different circumstances. First, we're going to take a toy example. And by toy, I just mean overly simplistic. So imagine that we have a Petri dish. And in that Petri dish, we have a substrate that supports bacteria. And I'm going to visualize that bacteria with that little green guy. Now, if we simply specify a few conditions, that resources that the bacteria consumes, like sugar, is continually added to the dish, and that on the basis of that bacteria's metabolic process, how it consumes the sugar and uses it, the bacteria in the normal course of events produces two offspring, two children, and then itself dies. Now, one more criterion that we're going to add, and what this is going to do is it's going to make sure that the population isn't growing exponentially, is that we're going to say that every generation, there's a 50% chance that one of the children is also going to die. And we can think of that as due to challenges in the environment, like maybe ultraviolet rays are coming in and disrupting their genetics. And then also, just to make sure that the screen doesn't get too overcrowded, we're going to do some other kinds of killing, but it's always going to be working against whatever hypothesis we're looking at. So those are the only things that are going to be going on in the following example. So here's our Petri dish and our first generation of bacteria. And just using those dynamics that I just introduced, we can see that every generation, we get reproduction and some death and reproduction and some death and reproduction and some death and reproduction and some death. So far, in this simple example, we have almost everything we need for natural selection to take place. The one piece that we're missing is the alternative. we only have a single example of a replicator. We only have one heritable design for replication. Now let's introduce an alternative. Imagine that a mutation happens. And what this mutation does is it causes a slightly less efficient metabolism so that fewer resources can be used to make offspring. In contrast to the original green type that's able to use the amount of resources it has available to produce 2 offspring, this new mutant is only able to produce 1 offspring. After a few generations, even if we presume that that mutant survived the first coin flip of death, that's not going to happen every time. And eventually we can see that this mutant will have been filtered out of the population. So that is a case of natural selection. We had alternative designs, and the design that had the best reproduction survived over time. But now let's consider a different mutation, this blue mutation. And this mutant is going to have the opposite effect. Where the green type, through its metabolism, produces 2 offspring, let's imagine that the blue type produces 3. Now, if we take our 50% death rate in reproduction and our 50% death rate, and now let's say that this is one of those times when an external event happens that causes additional death. And we're going to make that additional death affect the blue type more than the green type. I'm going to take 1 out of every 3 blues, but only 1 out of every 4 greens. Okay, so the blues did worse in that extra round. They reproduce and then die. Just moving them to the top of the screen now. They reproduce and then die. And again, let's just have another round of death. And again, I'm only going to be exposing the blue mutant to an additional death factor compared to the green. So in this case, let's say it's even more severe. Let's take 3 out of 5 blues and only 1 out of 4 greens. Even if their mortality rate is higher to that degree, we can see that over time, the blue mutant is going to be constituting a greater and greater proportion of the population. Such that eventually, with enough of those unlucky coin flips of chance, we're going to be left with only that blue mutation in the population. So what I want you to notice here is that this was a simple model and probably fairly silly. But to illustrate exactly what happened, it's helpful to think about how simple that effect was. All that second mutation did, the only effect that it coded for, was a marginal increase in reproductive rate. And because of that one difference, the mutation spread to fixate in the whole population. This word fixation refers to when an alternative gene spreads throughout a population such that its relative proportion makes up the majority of that population, or that the relative proportion stabilizes over time. And in this case, that stable point was all of them. Now, this wasn't driven by any outside intelligence selecting. The only process that was happening was that when designs were in the presence of a competitor that reproduced greater at a greater rate, that design that reproduced worse was filtered out, weeded out. There was less of a proportion of that design over time. And the important word here is relatively, right? Because when green was competing with red, green had relatively better reproduction. But when green was competing with blue, green did not have relatively better reproduction. So we can see that there's no inherent advantage, no inherent best. There's always the potential for a design that can reproduce better. Okay. We can, of course, model natural selection using different tools. This time, I'd like to propose a math model. In this model, we're going to think about a specific spot on a chromosome, or another word for that is a locus. And at that locus, we have two alleles. And allele is just a word that means a version of a gene. So at this locus, we have two versions of a gene that we can refer to as the P allele and the Q allele. And we can specify that mutation can turn P's into Q's and Q's into P's at equal probability. If we specify that capital P is defined as the proportion of P in the population, then we can think about an evolutionary algorithm that defines P in the next time point as P in the prior time point plus some effective mutation. And we can think about mutation as being a draw on a random variable. Here, I'm modeling mutation as distributed with a uniform distribution, which you can think of as boxcar shaped. It's as if I was drawing a number with equal probability from a distribution with exactly that shape. And so there was equal chance of getting a negative 0.06 as a negative 0.05, as a positive 0.01 as 0.06. Because this distribution is symmetrical around 0, the average effect, the expected effect, is going to be 0. But because of noise, and the big fancy word here is stochastic noise, random change, we do expect there to be changes in frequency, drift, random walk over time. So let's see what happens over 10,000 generations when the only things in the model are the prior state of the model and that randomizing effective mutation. If we start the population off at 50% P and 50% Q, we can see a random walk over time. And we ended up, in this case, with more P's than we started out with. But is that reliable? Is that predictable? Let's try another run. That one seemed to end up in a similar place. Let's try 8 more runs. As you can see, with a randomizing force of mutation alone, you can't have any prediction about, on the basis of where a population starts, how the distribution of genes in a population starts in its initial state, and where that population is going to end up over time. But contrast that to something where you could actually have a force of selection. So in this next model, I'm going to take the prior model and simply give P a slight advantage over Q. Just a constant 0.01 boost to its reproductive rate. So it's reproducing essentially 1% better than Q. We'll do this over another 10,000 generations. That's it happening 1 time. That's it happening 2 times. And that's it happening 10 times. What can we see in this example? All of a sudden, we're able to predict with much greater accuracy where the population is going to end up over time. We can see that there's going to be noise. We wouldn't necessarily be able to predict the difference between, say, 90% of the population and 100% of the population. Because of that constant rate of mutation, that frequency is going to be bouncing around over time. But with that constant force of selection, pushing it up, we can see that we have much greater predictive power over the population dynamics compared to a model that doesn't have natural selection. And last, let's look at a third way that we can model natural selection, and that's using what's called an agent-based computer simulation. This particular simulation, I coded in Java, but people code these in lots of different computer languages. In an agent-based simulation, the way that natural selection works is that you define how agents behave and what properties those agents have, how those properties are coded in their genes, and then you define an environment where, based on the properties that the agent has, they're exposed to an environmental circumstance where they can have an interaction. And on the basis of that interaction, they accrue resources. After that generation is over, if you set the reproduction of those agents, how many offspring they create, as a proportional to the resources they got, then that's a simulation that over time will simulate the evolutionary process. You would see natural selection operating given the replication of inheritable elements in the presence of competitors. So let's think about a particular example. In this model that I'm going to be showing you, we can imagine that there's an interaction type where 2 agents, 2 individuals meet. And when they meet, they have a resource like this sandwich. And that sandwich has a parameterized value, B. And the only choice these agents have to make is whether they're going to fight and pay a fighting cost or not. Now, we can think of 2 very simple strategies that we could code genetically to make this decision. And conventionally, these strategies are referred to as hawks and doves. When a hawk meets a dove, the hawk fights. In fact, hawks always fight. And doves don't fight. Doves never fight. And so we can think about what would happen if a hawk was willing to fight and a dove wasn't. In that case, the hawk would win the sandwich. Now, that's not going to be the only interaction type, of course. Sometimes hawks are going to meet other hawks. And in that circumstance, we might imagine that one of them randomly will win and the other randomly will lose. So one of them will get the benefit and the other will pay the cost. If that image is not clear, I was simply using Band-Aids to represent the cost that that hawk was paying. And so if we think about this interaction happening with a coin flip, then what hawks can expect is essentially the average of B and C. The third interaction type is when a dove meets a dove. And in contrast to the situation where a hawk meets a hawk, doves are not going to pay the cost of fighting. You could imagine that what they do is flip a coin and one of them gets the sandwich. And that would give you an average of each of them expecting half a sandwich for each interaction that they have like that. You could also simply imagine that they split the sandwich because they're able to resolve the problem peacefully. So just putting what I just said into numbers, we can think about the interactions between hawks and doves. And sorry that the sandwiches are still there. They were supposed to disappear. But we can see in terms of the parameters that we set what the payoffs would be. I'm coloring the payoffs to the row player. So this hawk and this dove, those are what's listed first, and those are in black. The payoffs to the column player are in blue, and those are listed second. So what I'm showing you here is that when a hawk meets a hawk, they both have the same expected outcome. They're both expecting to get the benefit half the time and to pay the cost half the time. And so their expected payoff in every round we can define as B minus C divided by 2. When a hawk meets a dove, the hawk is always going to get the benefit. And the dove is never going to get the benefit, but they're also never going to pay the cost. So they're going to get 0. And that's symmetrical to the case of where a dove meets a hawk, where now this dove is going to be getting 0 and this hawk is getting the benefit. And finally, when a dove meets a dove, whether you think about this as every interaction they're splitting the sandwich, or every interaction they're flipping a coin and trading off who gets the sandwich, then in either case, what the doves can expect is half a benefit from each interaction. And what I'm going to do now is I'm going to show you just what would happen in a world where these were the only dynamics plus mutation, and we can specify what the values that B and C are. So I'm going to pull a simulation over, and if someone can just give me a thumbs up to make sure that that's visible. Can you see that? Okay, great. So in a hawk-dove simulation, like I just said, you can specify how large a benefit is and how large the cost is. In this simulation, I'm also able to specify how big the population is, how many generations it's going to evolve over. And then there's an additional toggle that lets us see simulation, see evolution happening fast or slow. In this first run of the simulation, I'm going to have the benefit be half what the cost is. So our benefit is 0.3 in value and our cost is 0.6. And you're going to see hawks represented in red and doves in blue. In this left-hand side, what this square represents, every dot represents an individual, and the blue ones are hawks and the red ones are doves. And then on the right-hand side, I'm plotting the proportion of doves in blue and the proportion of hawks in red over time, from 0 to 1 proportion, or 0 to 100% frequency. So in this particular outcome, what we saw is that when the population started at around 50-50, it didn't really move from that particular equilibrium. And equilibrium is just a place where the payoffs to the two strategies are the same. And so they don't change in frequency. They return to that frequency over time. Now let's think on the basis of our model why we would expect this to be the outcome. Well, imagine that the world has a lot of hawks in it. If the world has a lot of hawks in it, then we're essentially in this column for the row player. And what we're looking at is hawks getting B minus C divided by 2 and doves getting 0, because hawks are almost always meeting hawks and doves are also almost always meeting hawks. That's going to be the interaction type that would dominate in a world that was mostly made up of hawks. If we specify that fighting is costly, in this case, fighting was twice the cost of how big the benefit was, this term is going to be less than this term. That is, doves are going to be doing better. And so if doves are doing better in a world with hawks, then in a world where hawks are a high proportion, that proportion is going to decrease over time. And the proportion of doves is going to increase over time. But let's now imagine the opposite world, where almost everyone is a dove. In a world where almost everyone is a dove, now this is the column that defines most of the interactions. A hawk meeting a dove would get the benefit, but a dove meeting a dove would only get half the benefit in expectation. And B, no matter its value, as long as it's positive, is always going to be greater than B divided by 2. And so parallel to the case, or symmetrical to the case, where if hawks have high frequency, it's better to be a dove, we would now see that if doves have high frequency, it's better to be a hawk. And so we have a force of selection that flips around. As the red line goes up, natural selection pushes it back down. And as the blue line goes up, natural selection pushes it back down. As the blue line goes down, natural selection pushes it back up. And as the red line goes down, natural selection pushes it back up. Let's see what happens when cost is cheaper. Let's say that cost is only a little bit greater than the benefit you could get. Notice that we got, in some ways, a different outcome, but in some ways, a very similar outcome. It's different in that stable level around which mutation kind of kept, or natural selection kept, even though mutation kept pushing around, the frequency of hawks and doves, that stayed in a frequency range where there was both in the population at a particular frequency that was higher than you would expect if it was the case like the prior math model, where there was only a single direction of selection. The difference is that the exact frequency at which that happened is different. Now, in a world that has less costs related to fighting, this world allows a higher proportion of hawks to be the dominant strategy. So we would expect when fighting is less costly, there to be more fighters. Think for a second about a prediction. I want you to predict what'll happen if I move cost to be less than the benefit. I'm going to give you a second. Think about what these payoffs would be. Try to make a prediction in your mind. Now, remember in your prediction that we shouldn't expect to see 100% or 0% the whole time, because mutation is always going to be moving things around a little bit. But what should we expect in this case? Now what we can see is that except for a little bit of mutation that is pushing the strategies around, we no longer have the direction of selection flipping around. Selection in this case is always pushing the red strategy up and the blue strategy down. Now let's think about a world instead where costs are high and I'm going to make benefits non-existent. Think about what you would predict. If this model has zeros in it for all of the benefits, but this cost for fighting is still substantial, you still pay a cost when you fight. Now we see relatively that opposite circumstance. Now we see it's relatively the case that doves are the dominant strategy, and that it's only mutation that is moving hawks around, or that final proportion around 100%. Mutation is constantly pushing it off 100%, but natural selection is pushing it towards 100% the whole time. So the concept that we're seeing here is that selection can be either directional It can always go in the same direction, or it can be frequency dependent. The idea of frequency dependent selection is that as the frequency of a strategy changes over time, the direction of selection is different at different levels of that frequency. And what we can say about the Hawk-Dove simulation is that as long as the benefit is greater than 0 and less than the cost, these strategies will be in frequency-dependent selection, and that we're going to see them being pushed to what's called an internal equilibrium, an equilibrium that's internal to the range of 100% and 0%. All right. So there's a few things that we can say now that we've seen several examples of how natural selection operates. First, natural selection is not the same as mutation, right? We saw that mutation happens. We saw mutation in each of our 3 simulations, and mutation does change the frequency of replicators over time. Mutation is defined as a random change in replicator form, such that your prediction about the relative frequency over time, except when you're reaching a boundary, is random with respect to the underlying design. In contrast, natural selection is defined as non-random changes in replicator frequency. Those can be directional changes. Those can be frequency-dependent changes. But as long as those changes are predictable on the basis of the dynamics, then the existence of natural selection allows us to make predictions that are quite accurate about the future state of a population. Now, if we think about that first situation of the bacteria in the dish, we can see that this is a situation that requires variance. No natural selection was happening when we just had those green bacteria. And it's true that mutation is a source of variance. But it's not the amount of variance that is important for determining the outcome. In fact, comparatively, the power of selection in determining the outcome can be much stronger, even when you're thinking about the differential in reproductive rate between the 2 designs, or the differential in how successful they are at gathering resources. If that differential is even quite small, we can see selection being a powerful force in shaping the frequencies of replicators in a population over time. So the idea behind incorporating natural selection into our thinking is that from this simple principle, this simple, you can almost think of it as a physical law, that replicators that change, that have a relative advantage in reproductive rate, because of that differential filter, will have a directional change in their relative frequency over time. That's going to happen. We can see that that's just the predictable outcome of any system that has those dynamics. And so even though that is a simple principle, we can say that it's actually powerful, because knowing this and only this, we can make predictions. But A problem that we face is that we're not robots that are making predictions. We're humans. And the human brain, as we'll come to learn, was not designed to think about natural selection. Our human brain has lots of intuitions that feel much more easy to make that actually go in the opposite direction of what natural selection would predict. And so we have to retrain our mind to be able to make these predictions accurately. It's often hard to think through the dynamics of natural selection because we have all kinds of intuitions that can get in the way. We have intuitions about right and wrong. We have intuitions about what's nice and what's mean. We have intuitions about what makes us happy and what makes us sad. When you're in doubt about what the prediction is, the way to make sure that you're making the right prediction is to return to that simple first principle, that replicators that cause their own reproduction better than competitors will come to predominate in a population over time. So let's see how you do. And I like to refer to this exercise as debunking the myths around natural selection. And we're going to go through several thought experiments. Imagine that there's two designs in competition, two replicators that produce some effect, and they're different replicators that have different forms that produce different effects. And so we have the conditions such that natural selection could operate. And the question is, what is our prediction about the design that should have the advantage? Which one would we, if we checked in on this population 1,000 years later, which of these two designs would we expect to still be around and the other one essentially gone? So let's imagine two organisms. And the question is, which would spread on the basis of natural selection? Let's think about design X, which has an easy life and is happy. And I apologize, this is covered up, but let me read it to you. Design Y has 2 children and a harder life and is relatively more miserable. So we have two differences here. We have a difference in reproductive rate, and we have a difference in overall life satisfaction. If we imagine that everything else is equal and that there's no additional knock-on effects, and as the parent of 2 kids, I can tell you that 2 is harder than 1, what would we predict? Would we predict that the one that has the easier life would be the one that spreads, or the one that has the relative reproductive advantage being the one that spreads? Going back to first principles, we would have to say the one that has the relative reproductive advantage. Natural selection doesn't care about anything, but it doesn't act as if it cares about comfort or happiness. And this is a common mistake that people make. This is a common myth that exists around how natural selection works. You can think of effects that a design could have, and those could be serving a reproductive goal. They could have an effect on reproduction. And there could be designs that produce effects that don't have an effect on reproduction. Effects like increases in happiness, or increases in your comfort level, or increases in your self-actualization, If you've taken psychology coursework, you might have heard of Maslow's hierarchy of needs. You might think of increases in your ability to solve that pyramid of needs. If those designs are not also producing a reproductive advantage, then that's not what's going to be selected by natural selection. In fact, to the extent that those impair reproduction, we would expect that they're selected out. So as we look around and think about the conditions that people are in, we shouldn't be expecting that natural selection is leading us to be happy. That's just not something that natural selection would do. Unless by virtue of being happy, that increased the relative rate of reproduction. Only in that circumstance would we expect designs for happy to spread. Let's look at another thought experiment. Imagine two organisms. One, X, in the course of its life, it lives for 80 years, and across those 80 years, it has two children. And compared to design Y doesn't live as long. It doesn't survive nearly as well as design X. And it similarly has two children. So we have two designs where one seems to survive longer than the other. What would we expect to happen if we checked in on these populations, on this population in 1,000 years? Now, you might expect, just what on the basis of your intuition, that the one that survives the longest would be the one that you would see. But think about what would happen at the end of 160 years. So if in the first 80 years, design X produces 2 copies, in the second 80 years, it's going to end up with 4 copies. At that time, after 160 years, design Y will have had 4 generations. And so after 40 years, it'll have 2. After 80 years, it'll have 4. After 120 years, it'll have 8. And after 160 years, it'll have 16. So even though X has a survival advantage, Y has a relative reproductive advantage. If we're thinking about designs for the longevity of life or the ability to survive, if that longevity of life or survivability doesn't have an impact on reproduction, or even negatively has an impact on reproduction, we shouldn't expect natural selection to select for that trait. This is another non-reproductive goal that many people think that natural selection should select for. The idea being that natural selection should select for a long life. And we can probably attribute that false inference to one of the original writers on natural selection, Charles Darwin, because a phrase that Darwin coined is that natural selection is the principle of survival of the fittest. And when this concept is being taught in school, often, if the teacher actually knows what they're talking about, they don't think about defining that term survival for students the way that Darwin defined it. Because this is not the intuitive notion of survival or fittest. When we use the word survival, we tend to think first about an individual organism surviving, but that's not what Darwin meant. Darwin meant survival in evolutionary time. Does that organism and their reproductive basis, does that survive generation after generation after generation? And fittest was simply a synonym. What it meant was the one that reproduced the best. The one, if you define the one that reproduced the best as the fittest, and survival as the one whose replicators persist the longest over generations, then survival of the fittest is simply a restatement of our idea that replicators that cause a reproductive advantage stick around longer and tend to be high proportion in the population. But it's not about survival in terms of lifespan. Let's think about another example. Imagine two organisms where X has 8 kids, and because the group only has a limited amount of resources, by virtue of having all of those kids, there's not enough resources to go around. And so when you have X in the population, 50% of the group starves and they die before reproduction. They die before they're able to have any kids. And that includes X. So X is only able to produce 4 kids per generation because 4 of them die. In contrast, Y only has 2 kids. And because of only having 2 kids and the reduced burden that puts on the society, only 25% of the group doesn't have enough resources to live. And so you can expect Y to have, on average, 1.5 kids per generation. But think about that. If there was a mixed group that contained X and Y, and X is getting 4 kids per generation, and Y is getting 1.5 kids per generation, even though Y is having a better effect on the group, X is the one that's going to replace it, because X has a reproductive advantage compared to Y. That is, designs that sacrifice personal reproductive benefit for group benefit, all else equal, they don't get naturally selected. In fact, they get naturally selected against. The common mistake here is the simplistic version of what's been called group selection, that the size or health of the group is something that natural selection would care about. But if those traits come at the cost of personal reproduction, if they involve sacrifice of the individual, that's not something that natural selection would produce, all else equal. One more. Let's think about design X that has 4 kids of her own, and her sister has 4 kids. Whereas Y, again, is making a sacrifice. She's reducing her own reproduction, so she only has 2 kids. But by taking that sacrifice, she's able to really help someone else. She's able to help her sister have 12. Now, we haven't talked about this yet, but if these designs are heritable, if they're inherited from parent to offspring, and let's imagine they're like us, and we're going to talk about this in a little bit. If the chances that you get a gene from a parent is 50%, because they're only passing on half of their genes to each child of theirs, then your sister has a pretty good chance of having the same gene that you do. And if your sister has a pretty good chance of having the same gene that you do, her kids have a pretty good chance of having that same gene as well. If we're using that 50% rate, then in one generation, X can expect 2 of her kids to have the gene, and 50% times 50% is 25%, she can expect one of her sister's kids to have the gene. And so on design X, we would expect the output to be 3 children. Design Y can expect one of her children, half of 2, to have that gene. And again, 50% times 50%, 25%. She can expect 3 of her sister's 12 children to have that gene. And so design Y, the expected output is 4 children every generation. So X is producing 3. And even though Y is paying a personal sacrifice in the body that Y exists in, it's producing a greater number of offspring. And so we would expect Y to be the design that spreads over time. And so we would say that designs don't simply spread by being selfish, right? It's not that natural selection always selects for selfishness. Natural selection can easily, we can see, select for a design that helps when the target of that help is kin. But does it always have to be kin? We can imagine a difference between organisms that are social and asocial. Asocial meaning they don't engage in social behavior. Now, social behavior isn't always profitable, but sometimes it is, right? You could imagine a design that goes on its own, lives its own life, doesn't engage with anyone else. And by doing so, it's able to have 2 kids. But design Y is able to engage in trade. And if you're engaging in trade for mutual benefit, if I have something that you want and you have something that I want, by trading, we're both better off than we started. And so design Y, by engaging in this kind of social cooperation, is actually gaining benefits, then we would expect Y to beat X. In other words, we don't expect nice strategies only in the case where that niceness is directed to relatives. This is another case of that common mistake that natural selection should only select for self-interest or selfishness. We can see lots of mechanisms where social behavior can be beneficial. And whenever that kind of nice social behavior is beneficial, if we can think of how that produces a net reproductive benefit compared to an alternative, that's going to be the design that we would expect natural selection to select for over time. Coming back to our definition of natural selection, there's one component that we really haven't dug too deeply into, and that's that replicator thing. What is a replicator? Getting this definition right is the key to understanding the class of things in the world that natural selection would apply to. For natural selection to apply, a replicator is anything that through its own interaction with the environment produces reliable copies of itself. So on that definition, let's think about a few possible objects that natural selection could operate on. Would natural selection operate on a copy machine? Copy machines certainly make copies, but copy machines don't copy themselves. They don't produce self-copies. They copy documents. So that's not something that natural selection would operate on. How about a species? Species certainly, they persist over time, individuals get reproduced. But is that collection of individuals the exact same after death and reproduction? Do species, do their genetics drift? Do species make more species that are reliably the same thing? No. How about an individual? In particular, a human, which is a class of organism that has two parents, where the parents engage in sexual reproduction. So every offspring doesn't have all of the genes of either parent, but only half the genes of either parent. Are you exactly like either of your parents? Did either of your parents make a self copy to make you? No, we don't clone ourselves, at least not yet. How about a gene? A little piece of genetic material that exists in one individual. When that individual reproduces, even in a sexually reproducing species, does that gene make copies that, because of that reproduction, can persist over time? Yes. That is our definition of self-copy. In fact, if we think about survival being the survival of genes over evolutionary time, we can think of some genes as being extraordinarily fit. There are copies of genes in us that existed more than 500,000 years ago. Sorry, more than 500 million years ago, more than 500 million years ago. There are copies of genes in us that date back to some of the first replicators that were creating cells nearly 1 billion years ago. Those genes have had incredible survival. They've produced an amazing lineage of copies that have survived a very long time. But what exactly are genes? Let's, for a bit, get more specific about what we're talking about, just to see that it's not magic. We're not giving natural selection any kind of magic ability. We're not allowing it to operate on a magic set of material. It's just the physical output of a biological and chemical system that has reliable effects. So you've heard the word gene before, and some of you might have taken some genetics coursework. But just so that we're all on the same page, let's think about what those genes are made of. So genes are a sequence of what's called nucleotides, where nucleotides are small molecules that are paired together into strings. And those strings of nucleotides are arranged into chromosomes. That long string has a start and a stop as a piece. And those chromosomes contain sequences that, by virtue of their interaction within the cell, produce effects. In fact, specific sequences of those nucleotides code for amino acids. And there's a collection of different amino acids. And when those amino acids are assembled, they turn into proteins. So let's think of that as protein X. Now, this is essentially what most people think of when they think of genes. But genes are actually more than simply those sections of nucleotides that have the effect of being transcribed into an amino acid and therefore coding for a protein. Because it's not simply like the cells create ingredients that just because the interaction of those ingredients, you get an organism. It's not like it's a recipe for baking a cake where the recipe is only a list of ingredients. If you wanted to actually bake a cake, you couldn't simply just take all the ingredients and dump them in a bowl. That doesn't go from ingredients to cake. To go from ingredients to cake, there also has to be a process. There has to be behavior. There has to be control, sensation, conditional design. Do this only after this happens. And so when we're talking about genes, we're talking about more than simply those portions of the chromosome that code for proteins. But we're also talking about specific sequences of those nucleotides that act like the logic. It acts like the same thing that you would code into a computer program, like start doing this if that's happening, stop doing this if that's happening. You could think of this like if-then statements in a computer language. And in fact, not all of these control effects happen strictly within the nucleotides of the chromosome. That is, it's not simply the code as one level of organization, but there's extra molecules that can be attached to those chromosomes that create an extra hierarchical layer of control. An example of this is methylation. So methylation is when a methyl group gets attached to a particular spot on the chromosome that causes the chromosome to get wrapped tightly around a histone molecule. Now, when the chromosome is wrapped up like that, the chemical, the transcriptase enzyme that comes in and reads the DNA to turn it into an amino acid and eventually a protein, it can't get in to read that. And so when that methyl group is attached, it basically turns off that section of chromosome. When that methyl group is taken away, the DNA unwinds, and now the transcriptase can come in and read it. And so we can think about this as like at the level of the DNA, we have one level of functional, like if you're used to, say, object-oriented programming, you have a sub-level of objects that have their own conditional logic and their own functions. And then you have an additional level of hierarchical coding, like there's a class structure where there's a superorder class. And that superorder class has additional logic that specifies when functions are called and when they're not. And even more, even if we simply have this design in our model of genes, that's still not complete because we have genes that are outside of the nucleus of our cells. For example, our cells have mitochondria in them. Mitochondria are little organs within our cells that are the pieces of our cell that produces the energy that the cell uses. Now, that mitochondria has its own DNA. The design of the mitochondria is produced by genes in the mitochondria. Those genes are not the product of the genes in the nucleus. And so we need to think about the effects on replication of genes in different places and how they can act on control signals based on where they're located. We're going to see the implications of that in a few more lectures. There's a few effects that I want you to just know about. So let's take a look at the codons, triplets of nucleotides that code for a specific amino acid, because I want to show you a few interesting properties. So what this graph is showing, and it's probably way too small for you to actually read. I don't care if you read it. All I want you to see is the color. Okay. So in different columns, in different rows, so on the vertical axis, we have the first nucleotide in the triplet, whether it's a T, a C, an A, or a G. And across columns, we have the second nucleotide. And so whether that second position is a T, a C, an A, or a G. And then within each block, we have the third nucleotide, whether that third spot is a T, a C, an A, or a G. So notice that there's no ambiguity. That is, there's no options. It's not that if you have a T, and a T at that spot, there's a choice of what gets produced. It's going to be phenylalanine. But notice also that you could have mutations that don't have any effect. In that first box, in the upper left-hand corner, notice that you have 2 reds and 2 oranges. That is, if you had a mutation in that third slot, it doesn't necessarily flip a red into an orange. You could have a mutation where it stays red and a mutation where it stays orange. That is, these could be silent mutations. It could be even if you and I had different nucleotides in our codon in that gene, we would still have the exact same effect. So that mutation would just be completely invisible. But on top of that, you can think of some of these amino acids as being substitutable for each other when it comes to making a protein, because ultimately that protein needs to have an effect. And sometimes it doesn't matter whether you have one amino acid or another, the protein is going to have the same effect either way. And so again, those mutations wouldn't eventually have an effect on relative reproductive rate because they don't have any substantive effect on how the protein operates. So what this means is that, yes, mutations happen, but it's not the case that every mutation in your DNA will actually have an effect on how your body is built or how your body acts. All right, let's zoom out now to the level of chromosomes. Typically, developing humans have 46 chromosomes, and it's not 46 uniquely different chromosomes, but it's actually 23 diploid pairs. Diploid means 2 copies. Half of those we get from our mother, half from our father. And there's another organization where we call 22 of these pairs, the ones that are numbered, the autosomes. Autosomes are equally likely to end up in bodies that are male or female. And that last pair of chromosomes, the X and the Y, are called the gonosomes. They're the ones that are not equally likely to end up in the bodies that are male and female. How does that work? Well, that Y chromosome, that tiny little Y, it carries a particular section of DNA that codes for a protein that has the effect of being an antigen that produces an effect that triggers a cascade of changes. And so what you have is 2 bodies that have essentially the same genes, just the presence or absence of this one gene, One of those bodies develops as a female, the other body develops as a male. And this is why the presence of that Y gonosome typically results in a male phenotype, in a male body. Phenotype is just a fancy word for the body that an individual has, where genotype is the fancy word for the set of genes that an individual has. But there's nothing that is exactly inherent in that Y chromosome producing that effect. It has that effect by virtue of the chemical sequence that happens. You could have a Y chromosome, and because of other chemicals in the system, the effect of that chromosome gets blocked. And if the effect gets blocked, then you could have the Y chromosome, but develop into a female body. And you could have other chemicals in the system that mimic or copy the effects of the Y antigen. And that means that you could have the case where you don't have a Y chromosome, but still end up having a masculinized or male body. And so that's why I say typically results, because it doesn't always result. Let's go back to this idea of epigenetic control. Epigenetic just means outside the genes. There are lots of ways that that can happen, but the one that I think is particularly interesting is, again, this idea of methylation. So this is an image of a section of DNA where those white spots that are in between the blue clumps, those are available for a transcriptase enzyme to come in and read them. In contrast, if you add those methyl groups, you get that DNA additionally wound up, and so they're not available, basically turning those genes off. The really interesting thing is that parents, when they pass on a gene, can methylate that gene differently. And so there might be the exact same gene that I got from my father and that you got from your mother. And the gene, in terms of the chromosome, is exactly the same. The sequence of nucleotides is exactly the same. But if fathers turn that gene off and mothers leave that gene turned on, it would be producing a different effect in you than it is in me. Or let's say that I got the copy from my mother, and for the girls in the class, imagine that you got that copy from your father. If I got it from my mother, I'm not going to be passing it on in that same form, because I would be a father and I would be changing how it's methylated. And so my children would now be getting the paternal or father form. Whereas for the girls in the class, even if you got it from your father, you would be getting it in the paternal form, father form, but you would be passing it on in the maternal form. This gives you this really interesting class of effects known as parent of origin effects. And if you can't yet imagine why natural selection might cause this kind of design, imagine that mothers and fathers don't always have exactly the same fitness interests, that what helps a woman reproduce and what helps a man reproduce are not always the same thing. And to the extent that those interests are different, that sets up selection for parents to have a different effect, for selection to select for a parent to induce an effect in the genes based on their sex, as opposed to based on the species that they're in, or the relative proportion of people in a population. So putting this all together, what can we say? We can say that genes are sequences of nucleotides that either code for proteins or control how and when those proteins are produced. And these proteins then interact to create the organism, your phenotype, your body. And ultimately, that organism, you, reproduces, and because offspring inherit a portion of the parent's genes, for humans and other diploid sexual reproducers, half of their genes, those genes have a chance to be reproduced. This is what it makes a gene count as a replicator. In fact, a gene can only spread, the only way that natural selection can operate on it is by that gene controlling or coding for proteins that cause that organism to produce better or worse than alternatives to those genes. How could a simple protein do this? How could there be a single change that natural selection could see such that natural selection could act on it? Let me give you an example that you do not have to pay attention to the specifics. I just want you to think about this example kind of generally, because we're going to come back to it later. And this is the example of the phenylalanine hydroxylase gene. Don't worry about it. The phenylalanine hydroxylase gene has one effect. It turns one amino acid into another. It starts off, if it takes phenylalanine, it turns it into a different amino acid called tyrosine. Why does that matter? Well, tyrosine is important. Our body uses it to create melanin for our immune system and to make different kinds of hormones. And actually, having too much phenylalanine is bad, because when proteins are being created, in particular when neurotransmitters, the chemicals in your brain that allow your neurons to signal to each other, if you have too much phenylalanine, it interferes with the creation of effective neurotransmitters. And so this gene, the gene that codes for phenylalanine hydroxylase, the presence of it increases reproduction. It has this positive effect on increasing the availability of tyrosine, and it has this positive effect by decreasing the availability of phenylalanine. And we can actually see that it's effective because people that have mutations in this gene get a disorder called phenylketonuria. The way this works is, well, we are diploid. We get copies from both parents, and often everybody has 2 copies of this gene. And if you have a mutation that gets passed on from one parent, that's okay, because you still have a copy that still produces that enzyme. You kind of have a backup on the other chromosome. But if you have 2 defective copies, that means you're not producing that enzyme at all. Now you can't make that enzyme. And so you have a buildup of phenylalanine and you have a deficit of tyrosine. In fact, this might be more common in your life than you might think. If any of you are soda or pop, depending on what you call it, drinkers, if you look at a can of, say, Diet Coke, you'll notice some language on it that says, attention, phenylketonurics contains phenylalanine. And the reason why it's interesting to see that is because that actually shows the effect of that gene. If you have the absence of that gene, if you don't have the phenylalanine hydroxylase gene such that you have PKU, you need to pay attention to what you eat or drink in this case. Okay. So to be selected by natural selection, that gene must have some effect. Like, for example, code for a protein that does something better than what some other protein can do, which is coded for by some other gene. And this is the effect that would cause that first gene to spread via natural selection. But like we saw in that PAH example, and the reason why I introduce it right now, is that the effect of phenylalanine hydroxylase depends on the rest of the genome, right? Because if you have a genome that overproduces phenylalanine and underproduces tyrosine, that's the genome that this mutation would benefit in, or not the mutation, that where this gene would find a benefit. If there wasn't a genome that produced too much phenylalanine and too little tyrosine, there wouldn't be any natural selection for this gene. And so we have to think about the effect of a gene in combination with the rest of the genome. Now, what this creates is what's you can think of as a double-edged sword. And in English, the expression a double-edged sword is used to mean that there's like a sharp edge that you can use for cutting, and so it's useful, but there's also this other sharp edge that might cut you. It's dangerous. So it's both useful and dangerous. That's the idea. And the use is that we can think about genes that through their interaction build complicated traits. But that also means that the effect of genes can be interfered with by the rest of the genome. And this should give us some caution when we're interpreting cases where it seems like a gene is causing a misfunction. What do I mean? Well, there are plenty of cases where we can tie a specific mutation to a specific disorder, like in that case of PAH, where mutations caused the disease PKU. Another example that more people know about are the mutations to the BRCA1 gene and BRCA2 gene that have been linked to an increase in breast cancer risk. And that connection is a really well-established fact. There's genetic tests that people get to test for those mutations. And if they have those mutations, they're in a very different risk category for breast cancer, and they're treated medically very different. But what do those genes do? And how should we think about what they're for? Because the mutations result in breast cancer, does that mean that the gene is a gene for breasts? That's probably what it intuitively would sound like, but it's not. If anything, the way that that gene works is that it codes for a protein that repairs DNA damage. And DNA damage is one of the ways that you can get a cancer. And so that's the reason why having this mutation allows cancer to exist and spread more easily. And so if anything, we can think of what this gene is as a tumor suppressor. And so we have a gene that does something very specific, resulting in a disorder that, based on the way that we think about it, that disorder happens in a particular area of the body, but that gene is not related to that body, or that body part, rather. It's a totally different effect. By an analogy, if you know anything about the mechanics of a car, you know that cars have many, many parts, and there's lots of those parts that you could break that would prevent the car from running. Like if you take out the serpentine belt, or if you take out the catalytic converter, or if you take out the battery, all of those would cause the car not to run. But given that you have a car, and given that you have, given that you have a broken car, and given that you have a particular brake, it's hard to tell exactly what that thing does. And to the extent that people have complex adaptations that are the product of many genes, that means that it's going to be hard to go directly from cases of dysfunction, cases of problem, to what genes actually do. The last thing that I want to introduce, and this is going to bring us to the end of the lecture, is I want to introduce this word adaptation. This is an important word. And so let's go back to our definition of natural selection to do it. If replicators that cause their own reproduction better than alternatives come to predominate in the population, then we can think of worse reproduction as the definition of an adaptive problem. And we can think of what natural selection selects for is the design that solves the problem. Those are the designs that are spreading. Those are the adaptations. We can think of this as like a lock and a key and how they fit each other in a specific way. An adaptive problem is any problem where when you solve it, it increases reproductive rate. And an adaptation is any solution to that adaptive problem. And thinking about it that way, we see why when we think about adaptations, which are the things that natural selection selects for, we have to think of them in the context of their adaptive problems. Those adaptive problems that occurred in the past, because that's the past where that natural selection could operate. All right, end of lecture. See you next week.